Yeah,
Today in lecture,
I got marked down at 1.5, and you got marked
we
down
have
at
2 parts. We're looking at the physiology of how a muscle cell contracts. And then we'll take a break and come back and look at the mechanics of creating a force of contraction. No. It was my fault this time, not the printers. I went back and whatever I loaded up, I must not have set the order button. So I asked for them to be here today. The latest will be here tomorrow. All right. But everything I'm saying today is written out in steps. So all of the functions I'm going through are printed sequence by sequence, step by step. So this is where we ended up on the Tuesday before break. And I want to do a little bit of review because we just finished the lab exam and I'm not sure how much of it actually penetrated into those empty crevices that you had emptied up by filling out the test. So what was our term for a single muscle cell? Remember the roots, the prefixes for muscle? We had 2 major prefixes that meant muscle? Myo and sarco. All right? So we use myo when we're talking about a muscle cell. So what's our word for a single muscle cell? A myofiber. There you go, a myofiber. All right? And so this is what is diagrammed in part here is a myofiber. So what is the name of the plasma membrane of a myofiber? The sarcolemma. Sarcolemma. Okay? Now, what is that? That sarcolemma is invaginated deep into the muscle cell to carry the electrical current into the muscle cell. Do you remember what that's called? It looks like a T. So its short name is a T-tubule. And that stands for transverse tubule. Now, what's the membranous series of sacs that are found inside the cell and wrap around the bundles of actin and myosin? The blue structures that you see. Sarcoplasmic reticulum. What's the ion that they store? Calcium. Good. What do we call, what's another name for actin and myosin, just as a general term? In our typical cytoskeleton, we call them microfilaments. So in the muscle cell, we call them myofilaments. And we call a bundle of myofilaments a myofibril. Okay. So that we're seeing now the muscle cell up on the board, and I want to look at a myofibril. Okay, so we're going to kind of take out this structure right here, pull it out. And there is no membrane around the myofibril holding it other than the sarcoplasmic reticulum. All right, so it looks like it's wrapped around with a thin membrane. It's not. It's just overlapping actin and myosin. And it's overlapping in a pattern. So the myosin is the thick filament. And associated with each myosin in an overlapping pattern are thin filaments, which are actin. So there are 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So they may share them with other myosin, but we have this pattern where they overlap. We can see both, okay? But they're in that kind of an overlapping pattern. So let's look at the structure of the sarcomere of a myofibril. So here's a myofibril. Here would be the red. What I'll be drawing in purple are the myosin filaments. And when we talk about a myosin filament, this is what we're talking about, all right? Multiple myosin proteins. So each myosin protein has a myosin head. But the myosin filament, the filamentous myosin, then has multiple myosin heads at each end. Right? So I'm going to diagram a pattern similar to that as we look at a sarcomere. So filamentous myosin, the myosin head. overlapping actin. And there would actually be 6 of these for each myosin. I'm just looking at a flat view. And the actin is bound by proteins to a disc-like structure. And then we would have actin bound to the opposite side. Overlapping the myosin as well. So on. All right, so I'm drawing this myofibril, but just looking at the surface of it rather than trying to make it three-dimensional. So as I draw this this is called a Z-disc. Or in this view, a Z-line, but it's a three-dimensional Z-disc. So from Z-disc to Z-disc, or Z-line to Z-line, this structure is identified as a sarcomere. Told you we're going to use sarco and myo so many times, it's like, what am I talking about now? So Z to Z. A sarcomere is the smallest individual unit of contraction in a muscle cell. So my elbows will be the myosin, the actin rather, attached to a Z-disc at either end, and that's the length of a sarcomere at relaxation. When the muscle cell contracts, the actin filaments are pulled towards each other in the center, and the sarcomere shortens. Okay? That's the essence of how a muscle cell shortens. The myosin filament does not get shorter. The actin filament itself does not get shorter in length. It's the distance occupied by the two. All right? So this, let's say that this would be a relaxed muscle cell, and if we were to draw a contracted muscle cell, it would appear more like this. Again, the same length of myosin. I'm trying to draw it the same length. I'm not going to bother with the heads. However, actin has been pulled inward. So neither the actin nor the myosin have changed length of filament, but the overall sarcomere is shorter. Okay? So how did we get to that point? Well, before we talk about that mechanism, let's identify some of the segments or regions of the sarcomere, and because these are in sync with other fibrils, the entire myofibril has the same pattern. We can see it externally. So Z-disc to Z-disc is our sarcomere. However, the actin here, between two sarcomeres that overlap, creates a light pattern, a light binding appearance. So if we look at an electron micrograph of this region right here, so here's the Z line, here's the other Z line. If we look at a micrograph pattern of that, you can see that where the Z line is lighter than where the myosin is. And our term for that light reflection is isotropic. So the area that is only actin, even though it involves two parts of two different sarcomeres, the area that is only actin is identified as the I band,
Which would be what part?
which would be this area right here. So what would this dark line be that's in the middle of the I band? The Z line, or the Z disc. Either or is fine, the term. All right, I band stands for isotropic, and it contains only actin. Okay. Now, the area of the myosin filament overlaps. The myosin is a thicker filament, twice the diameter of actin, plus it's overlapping with actin. So that contains a darker appearing region. So the width of the myosin filament is identified as the A band, which stands for anisotropic. And it's not only myosin, but it's the width of the myosin. So it's going to have overlapping actin to different amounts depending on whether the muscle is contracted or not. So it's myosin plus actin. But the width is determined by the length of the myosin filament. So this would be the A band right here. Okay? So I band, A band. But the sarcomere is Z to Z. So this would be the sarcomere. This would be the A band. This would be the I band. Right? Now, notice that in the A band, we have an area that is only myosin. That the actin doesn't overlap. And that is known as the H zone. So here, this would be the H zone. It's a little bit lighter because it's myosin by itself, right? So the H zone is myosin only. It's a subset of the A band.
Doesn't stand for anything other than H zone.
The dark line that runs through the H zone is due to proteins that connect each myosin to another myosin. And that's what you see right here. And so that is identified as the M line. Now over here on this diagram of a contracted muscle cell, which of these structures have changed in dimension with contraction? Is the sarcomere shorter or more narrow? What's the sarcomere defined as? Z disc to Z disc. All right? So here's Z disc to Z disc. Here's Z disc to Z disc. Is the sarcomere shorter? Because the actin moved towards the center. Therefore, the two Z discs move towards each other. And this one will move towards that one. So when a muscle contracts, remember we said the sarcomere is the smallest unit of contraction. So yes, it gets shorter. All right. So the sarcomere will get shorter. The distance between the two Z discs will become less when a muscle cell contracts. Will the I band become more narrow? Yes, I may not have drawn it so, but if I extend, so as the actin is overlapping more of the myosin, there's less free actin. Remember the I band is only actin. So let me illustrate. This time only this forearm is going to be actin, this one will be myosin. Okay? So myosin, the A band from here to here, here's the actin, and this will be part of an I band because it's overlapping. Muscle contracts. Is there the same or less free actin available? Less, because more of, so here we have free actin, double that for the other side of the Z disc, and now as it moves in, it's less. So during a muscle contraction, the I band will become more narrow as well. Because remember, the I band is not determined by the length of the actin directly, but by the amount of free actin. And so that amount that's not overlapping myosin becomes less. Because by definition, the myosin, the sarcomere becomes shorter as more of the actin overlaps myosin. Yes, Nadia?
If both sides of the I band or
The sarcomere?
being pulled, how does it go in?
They're being pulled towards the center.
But I mean,
So this one is being pulled towards the center, and all of these are going to do that. So this
it
one is
starts
going
in
this
the
way too.
middle somewhere?
Yeah.
Okay.
And they all go towards the center of the cell.
Okay.
All right, what about the A band? Does the A band become more narrow with muscle contraction? What determines the A band width? The width of the myosin. Is the myosin getting shorter? No. So neither does the A band. Okay, again, this shouldn't be something you memorize. You can figure it out. The A band, the myosin doesn't lose its length. It doesn't lose protein components. So it stays the same width. So does the A band. What about the H zone? What determines the H zone? The amount of free, non-overlapped myosin. As actin moves closer, that gets more narrow. Okay? So when a muscle cell contracts, the sarcomere gets more narrow, the I band gets more narrow, the H zone gets more narrow. The A band, the M line, and the Z disc don't change because those are determined by protein structures that aren't added to or removed from. The others are determined by overlapping, and that's what changes. Now, if we look at this image, or here's probably a few more, a cross-section through the sarcomere is going to show up different components. Again, this should make sense if you recognize what structures overlap. So if I do a cross-section through the Z disc, am I going to see any myosin? No. I will see actin, and because it's the Z disc, I will see the proteins that connect the actin to each other, like that. Okay? This diagram over here. Now, what if I'm looking at the I band? Just the actin. No, there is actually another small molecule called titin, and that's what you're seeing as these tiny little dots. Okay, titin, I'll get to it in just a second. Titin is a spring-like molecule that attaches the myosin to the Z disc. So I'll put it in there. I'll show you in just a second. Oops, wrong color. Did you get for having a green cap on a purple? So titin is a spring-like structure that attaches the myosin to the Z disc. And you can see that right here.
It attaches
the myosin to the Z disc. So right here. But so small it really doesn't become a visual part of our sarcomere when we see it in the microscope. So that's the A band. I'm sorry, that's the I band that we would see primarily just actin alone by itself. All right, now the A band has two parts, right? It has the part that has the overlapping actin and myosin, and it has the H zone, which is only myosin. So the overlapping part is going to have the big myosin filaments. With the smaller actin around them. And then the H band, running out of room here. The H band or H zone, where we're not at the M line, it's going to be just myosin. And then if we're at the M line, it'll be like this, where we have just myosin but we have the proteins. We don't have actin, but we have the proteins of the M line holding the myosin together. So if we look at this in the microscope, let me turn the lights off. This is a light microscope, and this is what we're going to be looking at in, actually I have an experiment to do on Thursday. Hope it works. I haven't done it in about 4 years. Stuff is coming in tomorrow, but it has to come fresh because we're looking at enzymes. We're going to take pieces of rabbit muscle that have been soaked in glycerin and cut them into 1 centimeter sections and then look at them under the microscope with or without salts and ATP. And if it works properly, we can actually see these changes in the I band, probably not the H zone, but in the I band in the light microscope. So with the proper ingredients, the muscle cell will be able to contract. And you'll be able to see that contraction in either measuring the length of your strand or in visualizing the narrowing of the I band. So here, if you look really, really, really carefully up close, you can see all the different structures that I listed. But even in the back, maybe, you can see the dark pink and the light pink with a line through it. You guys see that in the back? I've blown this up a bit. So the dark pink is going to be which component? That's the myosin. So that's the A band. All right, with overlapping actin. That's the anisotropic. The light pink is going to be the I band, and the dark line through the middle of that, it will be the Z. Remember, the I band has the Z disc. Okay, and if you look really closely, you can see a little tiny little bit of light in the middle of the A band. But I'm right up looking at it, and that would be the H zone. Okay, I don't see an M line in the middle of that. But the dark would be A, the light would be I, and so if we were to see muscle contracting, we would see the two dark lines in the I band closer to each other. All right, and so we'll probably have to use oil immersion to see this. We'll see how much we can see without, but that's what we'll be doing in lab on Thursday.
Cutting up the brain, you know what I mean? It looks like it should go